All right, so chapter 6 is way different than chapter 5. Chapter 6 is all about intermolecular forces. And before we get into that, we have to do some super duper basic review. How do we define a solid as opposed to a liquid? Yeah, it won't change its shape in general. So it retains a fixed shape and volume. It's pretty easy. The interesting thing with solid materials is they aren't stuck, right? The material, when it's a solid, can actually vibrate a little bit. That's generally how you feel that something's warm. If you touch it and it's warm, those molecules inside there are vibrating. So even though it is a static, stationary shape, it does vibrate a little bit. All right, so those are some good definitions. How do we define a liquid? Yeah, it fills the shape of the container. At least the bottom of a container. All right, and then last but not least, we've got gas. How do we define a gas? Yeah, it fills the entire volume of whatever it's in, right? So a liquid only fills the bottom. This will fill the entire volume of a container. All right, so if we look at all three of these, we said that solids vibrate, liquids tend to flow or move freely. And then when we get down to a gas, these move really fast. So really the big difference between a solid and a liquid and a gas is how quickly they move. A solid just vibrates a little bit, a liquid moves around, and a gas zooms around as a pretty general rule. All right, so we did a little bit of this with our water lab where we were trying to investigate converting water from its solid form to its liquid form to its gas form. What did you guys notice about the curve when you graphed it? Was it perfectly straight? What did it have? It fluctuated, right? Did some of you guys see kind of steps in there? Where it would go up and then plateau for a region and then go up some more and then plateau? That's pretty normal. And that's, every time you convert from a solid to a liquid to a gas, you typically go through this plateau region where the temperature doesn't change very much. And so let's talk about those changes. These are referred to as phase changes, anytime you convert from a solid to a liquid or a liquid to a gas or things like that. So melting is a pretty easy one to define. Melting is a phase change. What happens when something melts? Yeah, solid to liquid. And then we have vaporization. What happens with vaporization? Anybody else want to help? What's that?
Liquid to a gas.
Liquid to a gas. So you're making vapor. All right, and then we've got condensation. If you think about condensation at home, right, that's when you get moisture on your windows or moisture on the grass. So what do you think's occurring there? Yeah, gas to a liquid. Not too hard. And then last but not least, we've got freezing. With freezing, we're going from a liquid to a solid. So these are our four main ones. There's one more that we're going to talk about in a second, though. Is that what you were asking about? Yeah, there's one more, but these are the four main ones. So if we look at a graph, you guys might have had a graph that looks something like this for your water lab, right? So in this region down here, you're heating up your solid ice. So ice can be from 0 degrees Celsius to negative whatever, right? It could be negative 100 degrees Celsius. And you can slowly warm it up to 0, at which point it converts from ice to a liquid, right? And that's when you go through this plateau phase, right? Where you guys probably saw both ice and water. It wasn't pure ice, it wasn't pure water, it was kind of in between. And it probably hung out at 0 degrees for a while. Did everybody see that? Yeah. So it's pretty interesting that it kind of levels out there. And then after a while, all of your ice is gone, and then you're heating up that liquid water. You can warm it up or cool it down. And then you undergo another phase change where you start seeing boiling. That's where you get vaporization and condensation. Unfortunately, we didn't have tools for measuring the temperature of the gas portion, but you should have seen a nice plateau when it reached boiling. What was the temperature? 100 degrees, right? So you can go in this direction where you warm up your ice, you can melt it, and then you can vaporize it. Or you can go the other way where you can cool the vapor, you can condense it, cool the liquid, and then freeze the liquid, right? So that's the general water cycle. You can get rain from clouds that can hit the ground and then freeze. So that's the reverse of what we were doing. Does that make sense? I know a lot of this is super elementary. All right, there's one more phase change, though, that we haven't talked about. This is a special one. All right, this last one is where you go directly from a solid to a gas. Does anybody know what this is called? Sublimation. Sublimation. Not to be confused with the band, though. Does anybody have an example of something that sublimes?
Dry ice.
Dry ice. So dry ice is a good example of something that sublimes. Dry ice, if you have ever played with it, doesn't ever form a liquid, right? It just goes straight from the solid dry ice to gaseous dry ice. Unless you really manipulate the pressure. Is anybody here a coffee fan? Yeah, so I'm sure some of you guys have had decaf coffee before, right? It tastes awful, but some people like it. So with decaf coffee nowadays, in order to get the caffeine out, what they do is they take dry ice in a sealed vessel, a big stainless steel container, and under incredibly high pressures, they can actually turn that dry ice into something called supercritical CO2, where it will actually go through the liquid phase. Then what they do is they take that liquid CO2 and they percolate it through your coffee beans. And then after it goes through your coffee beans, it will settle down onto the bottom of the container. And then they release the pressure from the container. And what's left on the bottom of the tank is a bunch of caffeine. And your coffee beans are up high. And then what do you think they do with that caffeine? They do what? They sell it to Coke and Pepsi and other soda manufacturers. So supercritical CO2 is one of the weird exceptions where you do have to pressurize it a lot, but it leads me to my next point. All of these phase changes depend on pressure. So for example, if I were to go out into outer space and take water with me and then release the water out there, it would really quickly evaporate. Even though it wouldn't do that on Earth because we don't have pressure in outer space, right? So you have to take into account pressure. However, in this class, we're going to assume all our experiments are done on Earth under normal pressures, right? All right. So let's talk about the interactions between different molecules. And we have to use some kind of confusing terminology. These are called intermolecular forces, and it's not to be confused with an intramolecular force. So an intermolecular force is the interaction between two or more molecules. And the way I typically describe an intermolecular force is a molecule's stickiness. Some molecules tend to be really sticky. They like to really hang on to their neighbor. Other molecules just really don't care and they'll float around freely. So if we look at this example here, what do you think the molecule is that's shown? H2O, right? So H2O is a classic example of a sticky molecule. Up here we've got one molecule of water, and in the center we've got another molecule of water, and you can see all of these molecules are kind of hanging out close to that center one, and they're interacting in a non-covalent fashion, right? So an intermolecular force is a non-covalent interaction. So people get confused with this. Inter means between two things. Intra means between one thing, right? So a covalent bond, on the other hand, is an intramolecular force because it's within one thing. It's not between two things. Does that make sense? People get it confused a lot, but I always think of intrapersonal relationships, right? It's not how I feel about myself. It's how I interact with my neighbor. So you have to be very careful with the prefixes when you're describing these. All right, so let's talk about these non-covalent interactions in a little bit more detail. Whoop. I call these IMFs. Not the International Monetary Fund, but an intermolecular force. Typically, stronger IMFs lead to higher boiling points and higher freezing points. So these forces really impact the physical properties of a molecule. So let's take some examples of this. I liked using water as an example. And we'll compare this to methane, which is CH4. Methane's found in natural gas. A lot of new chemists are tempted to say, well, the heavier molecule should have the higher boiling point, right? So if we go through, we'd say, all right, water has a molecular weight that is really similar to that of methane's. It's about 18 grams per mole, where methane's is about 16 grams per mole. So same ballpark. One's not hugely different than the other, just a difference of 2. However, if we go and we look at the boiling point, what's the boiling point of water? Yeah, about 100 degrees, depending on the pressure. And methane, does anybody have a rough guess at what temperature methane boils at? I actually forgot to write it down in my notes, so I'm going to estimate too. I think it's like a negative 165 degrees Celsius. So even though they have a difference in molecular weight of about 2, water boils at 260 degrees Celsius higher of a temperature, right? So you can't just look at molecular weights when you're trying to analyze a molecule's boiling point. You have to look at the stickiness factor. Water is super duper sticky. It likes to hang on to water molecules. So when you're boiling it, it'll vibrate around, but it really doesn't want to get discharged and form a gas, right? Where methane doesn't really care about other methane molecules. It will get discharged and form a gas, no problem. So you have to think about it in terms of the intermolecular forces. So now what we need to do is we need to describe some of these intermolecular forces and come up with names to describe them. So I've got some cool examples. The first one has a bunch of different names. It's called dispersion, or you can call it van der Waals, or you can call it London dispersion, or you can call it an instantaneous dipole. Pick one that works. You don't have to create a big list. But I do like to show all of them because depending on the textbook or the source, you may see them used interchangeably. Typically, this is between two or more molecules that have no net dipole. So do you remember when we talked about Vespers theory? With Vespers theory, we said if you've got a net dipole, that's when you've got an imbalance of electron density towards one side of your molecule, and that depended a lot on shape. Well, it's coming back again. In this case, if you have a molecule that has no net dipole, that either means that all your dipoles are canceling out, or that you simply have no polar covalent bonds. Does that make sense? All right, now to get into the realm of abstract chemistry. So let's say we do have a molecule that's not polar in any way, shape, or form. We typically think of the electrons as being evenly distributed. However, if you were to take a camera, a theoretical camera, and you were to take a picture of a molecule, there's always a chance that an electron might be on one side of the molecule as opposed to the other, right? So just a snapshot in time. That will create an instantaneous imbalance of electron density. So if we look over here, at a snapshot in time, maybe the left-hand side of your molecule is a little bit more electron-rich than this right-hand side. We call that an instantaneous dipole. If there's another molecule nearby, there's also a probability of your electron-poor region of one side of a molecule instantaneously interacting with an electron-rich region of the other molecule. It's a really weak interaction, and it occurs very, very quickly. Yep?
Is that because you're taking the picture, or is that because there's a chance that it could be either one?
So it really has to do with the fact that electrons can't be everywhere at once. So in a molecule, the fact that you have electrons means there's always going to be a probability of your electrons not being evenly distributed, right? And because they're not evenly distributed, you'll always have one side of your molecule that's slightly more electron-rich than the other, and that can cause it to stick to another molecule that's also experiencing that phenomenon. So it is a little bit weird to think about it, but this entire intermolecular force is due to a momentary imbalance of electron density. It doesn't mean that you have a net dipole, it's just a snapshot in time. And I'll say it's usually thought of as weak. However, I really don't like that textbooks teach it that way because it depends. It depends on the size of your molecules. If you've got a ginormous molecule, those London dispersion forces or van der Waals forces can actually be quite strong. So I'll show you guys an example. We've got a gecko foot. If you look at a gecko foot under a microscope, you'll see all of these ridges and a bunch of fibers coming off. It's actually kind of weird to look at. If you go on Google, you can look at some pretty high resolution microscope images. But geckos can actually stick to walls through London dispersion forces. Their feet are thought of as non-polar, right? So they don't have a net dipole, but they don't want a net dipole because they want to be able to release their foot and then actually continue climbing. So it's a little bit weird to think about. And the main thing to remember with dispersion forces is they get stronger as surface area increases. So that's why a gecko's foot has all those ridges and all those little itty bitty fibers. Yep.
So can they control that dipole in their feet then, or is it just like random, or is it just weak enough that they can pull it off?
It's just weak enough that they can pull off their little pads and continue climbing.
So is there a chance, since there's a chance for it to be a dipole, is there a chance that it's just like they're not dipoles and it just falls off the wall?
Not really. They can't control it. It has more to do with the biomolecules that are on their little feet. It's weird to think about. The cool thing is that apparently the military's investigated gloves that are ultra sticky gloves that could be used for climbing. However, in all practicality, human beings are way too heavy for us to climb like Spider-Man. But it's still a neat idea. All right, so these are typically thought of as weak forces because it's an instantaneous imbalance of electron density. That leads us to the next one where we've got a net dipole. And these are called dipole-dipole forces. And with dipole-dipole forces, this is where you have an interaction between two molecules that have a net dipole. So in this case, it's not a momentary imbalance of electron density, it's a permanent imbalance of electron density. So let's take a look at some diagrams down here. And on the left, I've got a solid. And you can tell it's a solid because it's stacked in a really ordered arrangement. And on the right, what do you think that's probably going to be? Liquid. It's a lot less ordered. The molecules are probably going to flow a lot more freely. Okay, so if we go down and we look at these, you can see that, for example, on this end of the molecule, you've got an electron poor region. And over here, on this molecule, you've got an electron-rich region. Those two regions are going to be attracted to each other, right? Opposites attract. And then this occurs in a three-dimensional arrangement. So you can see down here, you've got another electron-rich region on another molecule. So you can't think of it as just two molecules interacting. It's really a big network of molecules interacting. And with solids, that's what leads to this really ordered arrangement of that brick and mortar stacking of molecules, right? With liquids, it's a little bit less ordered, but they still try to find that opposite to balance out their charge, right? So if you've got a partial positive region, that is always going to seek out a partial negative region to try to counterbalance itself. Does that make sense? All right, there's one special type of dipole-dipole interaction. Does anybody know the name of that? Comes up a lot in biology. So it's something bond. Anybody? Has anybody taken bio? A few people. Has anybody heard of hydrogen bonding in biology? Hydrogen bonding is a really special case of dipole-dipole. So we're going to spend some time talking about it. And I'm going to put bonding in quotes. So hydrogen bonding is a type of dipole that is very strong. Way stronger than a typical dipole-dipole. And the easiest way to describe this is that a hydrogen is shared between two atoms. Specifically, it's shared between a lone pair and a very electronegative atom. Does anybody remember where the most electronegative atoms are on the periodic table? The top right. Which one's the most electronegative? Fluorine. So typically, the closer you get to fluorine, the more electronegative you're going to be. So if we think about hydrogen bonding, there are three main elements that tend to stand out. Fluorine being one, it's the most electronegative. What do you guys think another one might be? Oxygen. And then a third one is actually nitrogen. So the way I tell people to remember this is ask yourself a simple question. Is hydrogen on the phone? So phone being fluorine, oxygen, or nitrogen. If you've got a hydrogen that's covalently bound to a fluorine, oxygen, or nitrogen, then it can H-bond. So let's take a look at this example. On the left, we've got hydrogen fluoride, which is HF. This was the molecule I talked about a little while ago that was in Breaking Bad. He used it to dissolve the body. It ate through his bathtub and then crashed through the floor. Pretty nasty stuff to work with, but it is like the world champ at H-bonding because fluorine's so electronegative. So this hydrogen over here is essentially being shared between this fluorine on the left and this fluorine on the right. It's essentially being handed off like a baton and nobody's letting go, if that makes sense. And then if we look down at water, we run into a similar situation with water, where this water molecule is here, this water molecule is here. The lone pairs on water, right here and here, are really, really attracted to that hydrogen. Which makes sense, because if we go through and we try to write in the net dipoles, I'm going to erase these. We said that oxygen is going to pull electron density away from that hydrogen. And then the net dipole for this other water molecule is going to be pointed up towards the oxygen. That leads to a very strong interaction between the oxygen and hydrogen that's referred to as an H-bond. The interesting thing is if you actually look at ice, right? It's impossible to tell which hydrogen belongs to which oxygen. They're being shared evenly among all of the water molecules. It's kind of weird to think about it that way. Does that make sense? All right, so let's talk about bio stuff, because I think hydrogen bonding is pretty neat. I kind of get carried away with this, so bear with me. Does anybody recognize the molecule on the left? DNA, right? So H-bonding is responsible for holding DNA together. We'll talk about DNA more at the end of the term, but I did want to introduce it. So if you've taken a bio class, you've probably talked about the double helix, right? You've got these two backbones that twist around, but the two backbones will stick together through hydrogen bonding of base pairs, right? So let's take a look at some common base pairs. We've got a G and a C base pair here. And if we look at this hydrogen right there, can you see that it's covalently bound to nitrogen? So the H is on the phone. And then over here on the oxygen, we've got a lone pair, which allows for a strong H-bond interaction to occur between that oxygen and that hydrogen. So if we go down one more, we've got another situation. Where down here, we've got a hydrogen that's covalently bound to nitrogen. And then over here, we've got a nitrogen that's got a lone pair. So again, we're going to get a second H-bond interaction. And then last but not least, we've got a lone pair right here. We've got a hydrogen right here. And we've got a third hydrogen bond. These are incredibly important for biomolecules. We don't want your DNA to just randomly fall apart. We want it to stay coiled together in nice packaged bundles, and then it will get read later on by specific enzymes. So some base pairs have 3 hydrogen bonds, others will have 2. It really depends. So let's make a note of this. Whoop. All right. Does anybody know any other important biomolecules where hydrogen bonding is involved? If it's not DNA, what's another important biomolecule? RNA. So RNA does have a lot of hydrogen bonding. Let's go outside of nucleic acids. What's another major class of biomolecules? Proteins, right? I heard somebody say it. So proteins are also super important. And hydrogen bonds are important for holding proteins together. Specifically, they hold them in special shapes. Does anybody know the different shapes that proteins will fold? Oh man, you guys are going to have to take more biology. It's a blast. It's beta sheets and, does anybody know the other one? Alpha helices. So proteins will fold into these complicated globs in specific patterns that are referred to as beta sheets and alpha helices. So proteins get held together. And they fold together in specific shapes. If you get a misfolded protein, you typically get very, very sick. So if you've studied genetic diseases at all, oftentimes that has to do with an enzyme or a protein not being made at all or being made incorrectly. So it's got some sort of defect and it doesn't fold in the correct shape. If you've talked about enzymes at all in your entry-level science classes, you can think of them as lock and keys, right? So your enzymes are the specific shape, and then the substrate's designed to fit exactly in the pocket in the enzyme. You can imagine if that enzyme didn't fold, and let's say it's contorted like that, it's not going to dock correctly with its substrate. So anytime you have a misfolded protein, it's a serious deal, and it's something that leads to oftentimes fatal genetic diseases. All right, there's also another weird thing that H-bonding does. It results in ice being less dense than water. Specifically liquid water. And people might say, okay, why is that such a big deal? Well, typically, you can imagine if you freeze something, it gets more dense than the liquid, right? Because it's getting more packed, it's going to be held closely together. That's what most liquids do, but with H-bonding, it will actually cause all of the water molecules to separate out a little bit so that they can adopt the best H-bonding configuration with their neighbor. So it actually changes the volume of ice so that it increases volume compared to the liquid. It's a little bit weird to think about it that way. But it's really important for our ocean ecosystems. You can imagine our polar ice caps would be very, very different if all of the ice sunk, right? What would be a major problem? Water level could rise. No polar bears that have nothing to stand on. There's also another really important thing. Has anybody heard of the albedo effect? A little bit? Are you in Sean's class?
Yeah.
Okay, so you might talk about that. So our ice basically acts as a big mirror, right? So sunlight will hit the ice on our polar ice caps and then get reflected back into space because ice is white, right? White reflects light. If all that ice were to sink, our planet would heat up super rapidly and it would be inhabitable for basically every organism. So it's very, very important that our ice floats, and it's kind of this weird phenomenon. Normally, the solid form of a compound is going to be the most dense, but for water it's a little bit different. So let's make a little note. Water is less dense in its solid phase. To allow for maximum Hbonding. All right, there's also another cool Hbonding feature, again going back to biology, but also something we've seen in class. This is called capillary action. So do you guys remember when we talked about meniscuses at the start of the year with the Penny's lab? Right, we said anytime you put a liquid in a narrow tube, it's going to kind of curve up towards the sides. That curve is due to hydrogen bonding. It's a little bit weird to think about, but on the side of these walls, we've got SiO2, that's glass. And this glass sticks to water and can pull it up. And the narrower it gets, the more it will pull up that water. And you see that curve as it's getting stuck to the sides, right? Why is this important in biology? Does anybody know? Where else is capillary action used? For capillaries, sure. Plants, right? If you water your plant doesn't have any sort of mechanism to pull up water. It doesn't have a pump or anything like that. Instead, it's all capillary action. So inside the plants, you've got a bunch of carbohydrates and biomolecules that are really attracted to water, and so that water in the ground slowly gets pulled up into the plant. So it's very important for plants to be able to utilize capillary action, and it wouldn't be possible without H-bonding. The other weird thing with capillary action is what occurs with compounds that don't hydrogen bond when you put them in a glass container. So what does Hg stand for? Mercury, right? So mercury is that liquid metal we talked about a few times. Mercury actually has an inverted meniscus because it doesn't want to be near water. It would rather be close to itself. So instead you get this weird phenomenon where the meniscus is actually going the other way. So it's kind of odd. Typically when we're using things like capillary tubes or graduated cylinders, we're using things that H-bonding, you tend to see that meniscus. All right, so one last one is that scientists use H-bonding to detect other molecules. So you may be wondering what the heck this picture is. Let's see if I can blow it up. This was actually what I made when I was in graduate school. And you can see in this molecule that I made, we've got a nitrogen right here, and there's a hydrogen on it, so that can hydrogen bond. And then down here, I've got another hydrogen that's covalently bound to a nitrogen. Same thing over here, same thing over here. And then up here, I've got a lone pair, but I can actually put on another proton up here, and then this can hydrogen bond. So I made a molecule that had 5 hydrogens pointed into a cavity, and they really, really want to find something negative to hang on to. So in this molecule, we made a sensor for chloride, Cl-, and it would actually get lassoed in the center and stuck into place. The cool thing with this molecule was when it found a chloride to hang on to, it would change color, and it would actually fluoresce a different color. So if you held it under a black light, it would glow. This actually didn't work out the way I wanted to at all. So what we were doing was we were trying to make a sensor for chloride, Chloride's involved in protein transport and a lot of important cell regulation pathways. It simply didn't work very well, but it worked well for nitrate. So does anybody know what nitrates are used in?
Gas or
Not gas. It's used in farming as a fertilizer, right? So if you've ever gotten Miracle Grow or lawn fertilizer, you'll typically see phosphorus and nitrogen contents listed on the bag. Those are essential fertilizers. So our sensor ended up being the best for nitrates. It wasn't very good for chloride. And then we were like, well, shoot, what now? And then we actually decided, hey, nitrates are pretty interesting. And so this is completely unrelated to the class. I'm more just bragging. So my old lab mate started a company where he made a sensor that would stick down about 6 feet into the ground, and it would tell you at various depths how much fertilizer was in the soil using our sensor. So even though we were trying to make a sensor for chloride, we accidentally discovered a sensor that was really good for detecting nitrate. And you may be wondering, why does that matter? Well, obviously, fertilizer is expensive. You don't want to waste money. What's another reason? Too much nitrogen can kill the plants. So if you've ever accidentally put too much fertilizer on your lawn, you've probably seen dead spots. Has anybody been down to the Gulf of Mexico? So the Gulf of Mexico has this big dead zone right now where there's like no fish living. All the animals kind of stay out of that region. And that's due to the fertilizer from the Midwest going into the Mississippi, going down to the Gulf of Mexico. And then what happens is the algae goes nuts. It says, we've got all this free fertilizer. Let's grow, let's grow, let's grow. And then they die. They go down to the bottom of the ocean, and then the bacteria says, all right, we're going to eat all of the algae that just died. But the bacteria soak up all of the oxygen, and then the fish don't have any oxygen to survive off of. So it's a really big ecological problem. And it's more of a side result of a failed experiment. So sometimes when reactions don't work, you actually get more interesting results out of it. But all of our sensors were due to hydrogen bonding, which was kind of neat. All right, so going back to on-topic stuff, let's talk about the relative strength of these intermolecular forces. Let's put weak up top. I'm going to put weak in quotes. What did I say the weakest one was? Yeah, dispersion forces like we saw with methane. So with dispersion forces, we said, all right, you've got these two molecules. One side of them is really electron rich due to a momentary dipole. The other side is electron poor. And you get this weak interaction occurring between them. So this is an attraction between non-polar molecules. Okay, so that's one end of the spectrum. And then we said after that you have dipole-dipole. And with these, you've got two molecules where there's a really big imbalance. In fact, let me draw it a little bit differently. Oh man, my artistry skills aren't on point today. All right, that's kind of kidney being shaved. And this region is really electron poor. And this region is really electron rich. Whoop. Because we've got net dipoles going in one direction. And then over here, we've got this interaction between two polar molecules. And remember, a polar molecule doesn't just have polar bonds, it has a net imbalance of electron density. So you have to have an imbalance. If you've got polar bonds, but they're canceling out in equal and opposite directions, it doesn't get counted. And then we said after this, we're going to keep on going down. We had Hbonding. And Hbonding, we said, is a situation where you've got hydrogen on the phone, meaning The hydrogen is covalently bound to oxygen, nitrogen, or fluorine. And in this case, this hydrogen is essentially being shared between the two oxygen molecules, or two oxygen atoms, excuse me. So it's a attraction between lone pair and a hydrogen that is bonded to fluorine, oxygen, or nitrogen. All right, there is one more. Whoops. Keep on going down. And this one's a little bit weird. This is called an ion dipole. And you can probably tell by its name. It's a situation where you've got a cation or an anion in solution. So I'll just pick one. So we'll just pick a cation, a generic plus charge. You can actually get an interaction between a molecule that has a net dipole, something like water. We know the oxygen side is really, really electron rich because oxygen is more electronegative than hydrogen. So all of these have a net dipole going this direction, right? Which is good because it helps stabilize that positive charge. The electron-rich region of each of the water molecules will surround the positive charge of that cation. So let's write this down. All right, and then last but not least, we have one last one. And these are the strongest intermolecular forces, and these are ionic bonds. And these are just simply attractions between cations or anions. All right, I think that's where we're going to stop today. On Monday, when we come back, we're going to talk about predicting intermolecular forces, and then we're also going to talk about solubility, too. And solubility has a lot to do with intermolecular forces. All right, remember, too, if you guys get stuck on sapling, you're welcome to email me. I'll hang out a little bit after class in the hallway, so if you guys have any questions, we can take a look at those.